Hello, and welcome to Chapter 5. Today we're going to be learning about membrane transport and cell signaling. We are also probably going to be joined by my lovely puppy Wesley, who is making a lot of noise today. So if you hear him in the background, just remember he's very cute. Okay, so we are learning about membranes, and the plasma membrane is the main thing that separates living cells from their surroundings. So it's the border, it's the, a lot of people confuse cell walls and cell membranes because we often think about walls as being borders, but that's just not true in living cells. It's the membrane that's the border. And the reason that it's the border is because, unlike cell walls, plasma membranes have this ability to be selectively permeable, or we can say they exhibit selective permeability. And all that means is that some things can go in and out of the cell much more easily than other things. And that's the nature of this membrane. It helps keep the living stuff inside the cell from anything that is not that cell. So we're breaking this down into parts like I usually do, and the first part of this lecture is the fact that we're going to learn about the various parts of the membrane. The membrane is an extremely complicated fluid mosaic of all kinds of things, including lipids, like we've already learned about phospholipids, and proteins. So phospholipids are the most abundant, like the most numerous of all the lipids in the membrane, although we are going to learn about a couple of other kinds of lipids as we go along. One of the important features about phospholipids is that they are amphipathic. And amphipathic is a special way to say that it contains two very different regions. And of course, you've already learned about this. Phospholipids have hydrophobic heads and hydrophobic tails. That's actually what makes it to be so useful in a watery, aqueous environment that most cells live in. Even the cells in your body live within the watery environment of your body. Your body is made up of more than 70% water. So water bathes both the inside and the outside of a cell, and it's that hydrophobic region in the middle that really helps with this as well. So they're both important. Let's take a closer look at those phospholipid molecules. Remember, we've already looked at this before in a couple other chapters. So membranes are fluid mosaics of our phospholipids and a whole bunch of other stuff, right? Proteins too. When I say the word fluid mosaic, it's really important that you understand what that means. I'll follow up with that on a future slide too. But basically, you can think about it as like an iceberg floating in the ocean, right? You've got this big thing. The icebergs don't tend to stay still in the ocean. They float around. So that's the idea of the fluid mosaic. I don't want you to confuse the fact that most of those proteins float around. Not all of them do. Some proteins and some lipids actually kind of stay in certain regions of the cell. But again, we'll get into the more details of that later. So back to this amphipathic nature of our phospholipid. The heads are hydrophilic. That means they have polarity, and they are attracted to the polarity of the water molecule. The hydrophobic tail regions, the regions that are mostly made up of hydrocarbons, are hydrophobic, and water is not attracted to them, and so they mostly just hang out together in the middle of this bilayer. So how do we know that the membrane is fluid? Well, we've done some very interesting research. Like if you take two cells, one from a mouse and one from a human, and you put them together, you kind of force them to merge, The proteins that are found on the mouse cell and the proteins that are found on the human cell actually do merge and distribute very equally around the cell, and that takes about an hour. Very interestingly, they also looked at the distribution of the phospholipids, and the phospholipids, they mix together much, much, much more quickly than the proteins do. So we do from this see that it takes a while for them to move, and we also see that some of those proteins seem to be anchored and some seem to drift more easily across the membrane. So even humans and other mammals that are able to maintain a steady homeostasis of temperature, they still have to be able to maintain fluidity even when temperatures change. So like we in part do that because we can maintain our temperature, but not everything can. Not like fish can't maintain their temperature very easily. So as temperatures cool, membranes can switch from a fluid state to a solid state. And the temperature at which the membrane solidifies depends on what kind of lipids are in it. The thing is, the membrane functions best when it's more fluid. So one of the ways that membranes handle dealing with lower temperature is to have phospholipids that have a lot of unsaturated hydrocarbon tails. Remember, we've already talked about what unsaturated means. It means that there's lots of double bonds, right? And so if you're looking at a phospholipid bilayer that has just tons and tons of saturated hydrocarbons, they pack together really tightly, and that becomes very, very viscous, or you could think about it as like thick. On the other hand, when you've got lots of double bonds present in those hydrocarbon tails, the phospholipids maintain some distance from each other. These are unsaturated fatty acids, and they have this bend in them, and that allows them to maintain their fluid nature in colder and colder temperatures. Another thing that helps with maintaining fluidity is the fat or the lipid cholesterol. Now, we tend to think about cholesterol as being a bad thing, but really, cholesterol is a very important part of the phospholipid bilayer. Lots of cholesterol means that in very cold temperatures, those membranes stay more fluid. And if you have less cholesterol, then you're less able to maintain that fluidity in cold temperatures. So again, fluidity is extremely important. Generally speaking, if you want to think about that, you can think about the fluidity of the phospholipid bilayer in a normal circumstance. It's kind of being like how you would pour olive oil out. It's thick, but it moves. Again, the two things that help maintain fluidity in cold temperatures are unsaturated hydrocarbon tails and cholesterol. Of course, some things have evolved in cold temperatures, and some things evolve in warm temperatures. Things that have evolved in cold temperatures are more likely to have those unsaturated fatty acid tails and higher levels of cholesterol. Things that have evolved in warm temperatures are more likely to have saturated fatty acid tails and have little or no cholesterol. Interestingly, organisms that live in environments where the temperature fluctuates a great deal they've evolved the ability to change the composition of their plasma membrane depending on what time of year it is or what temperature it is outside. Besides thinking about it as like a fluid mosaic, you could also think about the membrane as a collage of different proteins kind of just embedded in this matrix of the phospholipids. But all the proteins that we're going to talk about have very specific functions, and some cells are going to have more of one kind of protein, and some cells are going to have more of another kind of protein. This is just a generic discussion of what those proteins are. First of all, let's talk about location of proteins. The first location is integral, or some people say integral, but I always say integral. Integral proteins are proteins that go all the way through the plasma membrane. You can see an integral protein right here. And a lot of these are called transmembrane proteins, simply because they go all the way across. Transmembrane is another way of saying integral. If a protein is integral, if it's a transmembrane protein, then there's going to be regions of that protein that are hydrophilic. For example, the regions up here on the well, this is the inside of the membrane, it looks like from the picture, or it could be down here. Those again are going to be hydrophilic. The hydrophobic regions are the regions that are found in the center. And of course, this makes a lot of sense when you think about it, because if this region in the center here were hydrophilic, it wouldn't be able to stay in the hydrophobic region of the plasma membrane. If you look at it, kind of think about it at a deeper level, the kind of amino acids that make up this region of the protein are going to have those hydrophobic side chains, and the regions that make up this region of the protein are going to have those hydrophilic side chains in the amino acids. The other kind of protein are the peripheral proteins, and peripheral just means like on the side, right? So here's a couple of peripheral proteins here and here, and those are really just loosely bound to the plasma membrane, and they do serve important functions though. There are 6 different kinds of membrane proteins that we're going to be talking about, and I'm just going to move into the next slide to show you what those 6 are. You can read them here for yourself, but let's take a closer look at those 6. So starting out with the transport protein. Transport proteins are any proteins that span the membrane, but they have a very, very specific function, and that is that they allow substances to move across the membrane more easily. We're going to find out about how hard it is in a later slide to cross that membrane without a little bit of help. So transport proteins help other things move across the membrane. They might just flow through a process of diffusion through that transport protein, or they might go against their gradient and go away from, basically they're going away from where there's very few of them towards where there's already a lot of them, and that needs a little extra energy boost. That happens with ATP. The second function is the function of an enzyme. And an enzyme is any protein, as we've learned before, that helps with chemical reactions. There are proteins embedded in the phospholipid bilayer that help chemical reactions speed up. These enzymes grab onto the reactants and produce the products. And the cool thing about them being in the membrane here is that when 2 enzymes are right next to each other, like you see here, the product of one becomes the reactant of another. And so you get a nice little chain of chemical reactions happening extremely quickly. The third thing is signal transduction. And signal transduction happens when a receptor molecule or receptor protein that you see right here binds onto a signaling molecule and sends a chemical message from the outside to the inside. Now, it's really important to understand that with these signal transduction molecules, we're not doing transport. It's not happening like that. It's happening where just another signal kind of says, hey, I've just, I got this message, and I need you all inside the cell to do something different. That's what's happening. So an external messenger is which we call a signaling molecule, binds to the receptor. The protein changes its shape because it's bound to the signal, and it allows a message to be carried to go inside. Again, the molecule itself is not going inside. It's just the change of the shape of the receptor molecule that's causing the signal to go inside. The fourth kind of protein found in the plasma membrane is something involved in cell-to-cell recognition. So here's two separate cells, right? Two separate plasma membranes, and this protein is binding to this protein through this little bit here that's attached to the first protein. That's a carbohydrate. I don't know if you recognize, but those are little hexagons, and hexagons are almost universally the shape that indicates that you've got a carbohydrate there. If a carbohydrate is attached to a protein, we call that protein a glycoprotein. So this one right here is a glycoprotein because it's got the carbohydrate attached to it. One cell will recognize the glycoprotein of another cell, and that allows a signal to be passed. The next one is intercellular joining. We'll spend a little bit more time on this a little bit later in this chapter, but proteins allow two cells that are neighboring to bind to each other in intercellular joining. Those are we'll learn about gap junctions and tight junctions a little bit later in this chapter. Finally, we have those integral proteins here that are attaching to the cytoskeleton inside the cell and the extracellular matrix outside of the cell. So we might see proteins binding to microfilaments. They could be covalently or non-covalently bound to those membrane proteins, and what this does is this helps coordinate the activity of the cell. It also helps cells. It helps cells keep in touch with each other, as well as allow cells to be in touch with indirectly in contact with different parts of the interior of the cell. So I mentioned that we would talk a little bit more about those carbohydrates and how they're involved in cell-to-cell recognition. So it's so important, especially in multicellular organisms, that cells recognize each other as being the same or being different. And it's all about those carbohydrates that are found on the outside, or the extracellular. Let's just highlight that word for you, that this means outside of the cell. Extracellular surface of that plasma membrane. Those carbohydrates, again, are so important, but they could be either connected to lipids or proteins. If those carbohydrates are attached to lipids, we call them glycolipids. And if they're attached to proteins, and this is much more common, they're called glycoproteins. Those carbohydrates that are found on the outside of the plasma membrane are unique to each species, and it's unique to each individual, and it can help cells tell the difference between individual cell types within a body. When we get into talking about blood typing later in the semester, we'll talk about these carbohydrates again, in genetics too. We'll cover that. One of the things that we have been able to just kind of recognize based on these photographs or pictures that I've been showing you is that membranes have different sides to them, like we've been talking about inside and outside. And how they look are actually really different inside and the outside of that plasma membrane. Very, very different. For example, you're going to find the active site of enzymes. That's a term you haven't learned yet, but we're going to learn in the next chapter. The active site of those enzymes are going to be found on the inside, and we're going to see those carbohydrates, they're going to be more on the outside. This asymmetrical arrangement is really determined by our endoplasmic reticulum, right? And that's symbolized by ER, it's like shortened to ER, as well as the Golgi apparatus. And if you don't remember what those do, you might want to do a quick review in the last chapter. So just a really quick look about how the ER and the Golgi help determine the inside and outside. You can see the interior here of the Golgi apparatus. And when it buds off a vesicle, that vesicle might fuse to the plasma membrane. And when it fuses to the plasma membrane, it's going to release its contents. But you can see that the inside of that membrane, it looks more blue, is now the outside of the plasma membrane. And that is in part how our cells figure out inside from outside, how they're determined, how they get put the way they get put. So we're now done with the first part of chapter 1. I'm sorry, the first part of chapter 5. And we're going to move on to the second part, where we talk about really focusing here on the selective permeability and how things directly cross a membrane.